Hi, my name is Liga Fermasando, and today we are going to talk about the gene therapy and the cell therapy. The gene therapy is the introduction, removal, or the change in the content of the person's genetic code with the goal of treating or curing a disease. It is also the transfer of the genetic material changes how a single protein or a group of protein produced by the cell. The cell therapy is the transfer of the intact live cell into a patient to help lessen or cure a disease. The cell may originate from the patient or from a donor. Note that the gene therapy involves the transfer of genetic material, usually in a carrier or vector, while the cell therapy involves the transfer of cell with the relevant function into a patient. Both goal of the gene therapy and the cell therapy is to cure the fundamental cause of genetic disease and acquired diseases.
Hi, I am Christina Rinsorio and I will tell you about how does the gene therapy works. Recognizing this, scientists have been working for decades on ways to modify genes or to replace faulty ones with healthy ones, to treat, to cure, to prevent diseases or medical conditions.
Sometimes, whole or a part of a gene is defect or missing from birth, or a gene can change or mutate during the adult life. So any of this variation can disrupt how proteins are made, which can contribute to the health problems or health diseases. In gene therapy, scientists can do one of the several things depending on the problem that is present. So they can replace the gene that causes a medical problem to one that doesn't. Basically, they add the genes to the body in order to treat or fight the disease or turn off the genes that are causing problems. So, basically, our doctors and scientists are hacking the gene of a living human. So using genetically engineered retroviruses called vectors, in which the scientists infect human cells, and these retroviruses can be programmed to carry a gene or a DNA that will overwrite the messed up mutations and make it work properly. So gene therapy can be used to modify cells inside or outside the body. So when it is done inside the body, a doctor will inject the vector carrying the gene directly into the part of the body that has defective cells. While the gene therapy that is used to modify the cells outside the body, the blood, the bone marrow, or another tissue can be taken from the patient, and specific types of cell can be separated out in the lab. So the vector containing the desired gene is introduced into the cells. The cells are left to multiply in the laboratory and are injected back to the patient, where they continue to multiply and eventually produce the desired effect. In simple terms, gene therapy works by replacing a defective gene, changing genetic information with a population of cells for the purpose of the prevention of a disorder or alleviating the symptoms of a disorder. Gene therapy is still in tendency, but the promise of the future cares. I mean, from the cure of the cancers to genetic disorders, these things are incredible, and I, for one, cannot wait to see what this brings.
Hey guys, so the third question asks about the strategy of gene therapy, and it mainly specifies on somatic cell therapy instead of germ cell therapy, considering its ethical issues. Now, we will be talking about the three strategies. First, we have gene editing, then we have gene addition, lastly, we have gene-based immunotherapy. Gene editing targets a kind of molecular scissors called a nuclease to make a break in a particular gene. Simply, it means to break something in the DNA. There are two kinds of repair possible with its different effects. First, we have non-directed repair, which means the body itself heals the break in the DNA. Then we have directed repair, which means scientists give the body instructions on how to repair the DNA break, and the body inserts new DNA at the site of the break. Next, we have gene addition, which means it uses a delivery system called a vector to insert new genes directly into the cells. The vector used is a part of a virus, since viruses are capable of delivering genetic material into cells. The process is a vector is much like a delivery truck full of packages. Then the truck makes sure the genes are delivered into the cells. Insertion of a functional gene can take place either outside or ex vivo, or inside or in vivo in the body. Lastly, we have gene-based immunotherapy. It means adding a gene to the body's existing immune system cells that can help them to work more effectively against a disease. Example, presently, this approach is being developed to help immune cells, such as the T cells, to locate and destroy cancer cells, specifically tumors. And that's it.
Hi, my name is Beatriz Tobligan, and I will explain to you the uses of gene therapy. The uses of gene therapy includes the following. First, it restores missing or faulty genes in order to cure diseases and help the body build better immune system. Second, it delivers genes in a faster tempo for the elimination of cancer cells. Third, it supplies genes that causes cancer cells to regress to normal cells. The second half of the use of gene therapy will be discussed to you by Ms. Sarilla.
Good day everyone, my name is Christine and I'm going to add something on what Bea did discuss about the uses of gene therapy. So here are the uses of gene therapy. It delivers bacterial or antiviral genes in the form of vaccination. It also provides genes that promotes the growth of the new tissues. It also provides genes that stimulates for healing. In conclusion, we can say that gene therapy is beneficial, especially when we talk about human health.
Hello, I'm Sean and I'm going to discuss the different types of cell therapy. So these are the types of cell therapy. So first is the most common type of cell therapy, which is the blood transfusion processes, which includes red blood cells, white blood cells, and platelets transfusion. Another type of cell therapy is the transplantation of hematopoietic stem cells to create bone marrow, which is also called as the bone marrow transplant. We all have the immune cells therapy. It is a cancer treatment designed to boost or enhance the body's immune system to destroy cancerous cells. We have different types of cells used for cell therapy. The first type of cells are the pluripotent stem cells. The pluripotent stem cells are one of the cells that are self-replicating. These are derived from human embryos. We have 4 types of pluripotent stem cells. First are the induced pluripotent stem cells, the embryonic stem cells, the nuclear transfer embryonic stem cells, and lastly, the parthenogenetic. Another type of cells that are used in cell therapy are the multipotent stem cells. Multipotent stem cells are also one of those cells that are self-replicating. These are present in a specific tissue or an organ. Most adult stem cells are multipotent stem cells. These are the types of multipotent stem cells. First, we have the hematopoietic stem cells, the mesenchymal stem cells, neural stem cells, and the epithelial stem cells.